So now that we've gone into detail about the different pieces of the adaptive immune system, I want to talk a little bit more about memory, about immunity. So memory is the most important feature of the adaptive immune system. The first response, the first time you encounter a pathogen, is so slow, it's barely effective for some of the more serious diseases. By the time enough T cells and B cells and antibodies are produced that are capable of identifying and destroying the disease, you could be dead if the disease is fast acting. But if you survive it and the disease doesn't mutate its antigens constantly, remember antigenic variation for mechanisms of microbial pathogenicity. So if it doesn't mutate, if it doesn't have antigenic variation, then you will be immune to it for a long, long time. So first exposure, it takes 4 to 7 days before any measurable antibodies are being produced in response to infection. 10 to 17 days after infection is when you get your peak antibody production. So your adaptive immune system hasn't geared up to its full extent for about 2 weeks after infection, the first time you encounter a pathogen. Second exposure, it only takes 2 to 7 days to get to peak antibody production, and you start making antibodies pretty much right away. The memory cells are already there, living in your body. You have this whole army of B and T cells devoted to this one pathogen, so you can just skip the first steps of the adaptive immune response process. You don't have to generate those memory cells, those daughter B and T cells, right? They're already there. There are 4 styles of immunity. We have naturally acquired active immunity, naturally acquired passive immunity, artificially acquired active immunity, and artificially acquired passive immunity. Naturally acquired, both of the naturally acquired immunities just mean you don't need a lab or a doctor's office. It happens, it's been happening since the beginning of humans, since for all animals really. Artificially acquired means you need a lab. Active immunity means that you have living memory cells producing antibodies. You have living B cells that are actively producing antibodies. That's active immunity. Passive immunity means you're getting antibodies from someone else. You're not making them yourself, but you're getting them from someone else. Okay? So naturally acquired means no lab needed. Artificially acquired means you do need a lab or a doctor's office. Active means you're actively producing antibodies. Your own cells are actively producing antibodies. Passive means you're getting antibodies from another person who's making them, and you are not making them. So let's go through each of these one at a time. Naturally acquired active immunity is when a person gets sick, recovers from the illness, the memory cells give immunity for a few years or for a lifetime. Naturally acquired passive immunity, that's when a mother's antibodies are transferred to her infant, and that can happen through the placenta, and also it happens through breast milk. But this kind of immunity lasts only as long as the antibodies are being transferred. This is very important for protecting the infant before infant immune system develops. It takes an infant's immune system takes 5-6 years before it's fully developed. And during that time, children are more vulnerable to disease because they don't have a fully developed immune system. So during that time, in the womb and immediately after birth, a mother can give immunity to her child through her breast milk. But remember that means that the child's B cells are not actually producing the antibodies. They're getting the antibodies from the mother, but antibodies are not alive. Antibodies are just proteins, and they degrade in the body after a few weeks. So they really only last as long, this kind of immunity only lasts as long as the child is receiving those antibodies. Artificially acquired active immunity. So this is vaccines. So when you get a vaccine, you get a killed version of a pathogen or a living microorganism that is non-pathogenic but similar to a pathogen, or some inactivated toxin like a toxoid. So you take this non-pathogenic version of a pathogen or non-toxic version of a toxin, you expose the immune system to it. The immune system doesn't know that it's inactive because it doesn't, it's not conscious, right? So it still does its thing. It still does the whole generating this army of daughter B cells, daughter T cells. And so you still get that memory from it, but you can skip the whole getting sick part. So in artificially acquired active immunity, you've taken something that you've made in a lab, the mutant microbe that's not pathogenic, the killed version of the microbe, the inactive toxin, you've put it into someone's immune system, but it's the end of this is the same as naturally acquired active, where you then generate living B cells that are actively producing antibodies that destroy that particular pathogen. Artificially acquired passive is when you inject antibodies into a patient from an animal or from a person who already has immunity. An example would be antivenom. So if you get bit by a rattlesnake, you probably don't have immunity to rattlesnake venom, but you can get antibodies produced by a horse who's been exposed repeatedly to rattlesnake venom. That horse has B cells that generate antibodies that neutralize that rattlesnake venom. So you can take those antibodies out of the horse blood and inject it into a human patient to neutralize the rattlesnake venom in that person. Now again, just like naturally acquired passive immunity, this person is not actually immune after this to rattlesnake venom. You injected antibodies that neutralize the venom, but those antibodies degrade. They're not living. They don't stay in they degrade in the human's body, and the human patient does not actually have B cells yet to protect them from any future bites. So that's why it's passive immunity. The horse had active immunity, and you've taken the antibodies from the horse and given the antibodies to the person. The person does not have B cells that are producing those antibodies still, so it's passive immunity. And this, incidentally, this is also how we treat botulism. We take antibodies from another organism that has immunity to neutralize the botulism toxin. And also, if a person gets infected by Clostridium tetani and has tetanus and they haven't gotten vaccinated, then there's also antitoxins for that Clostridium tetani toxin. And those are just antibodies from another organism that was immune. So those are examples of how you would use, you get this artificially acquired passive immunity from a doctor's office. So we've been talking about all these immune cells, but we haven't talked about where they live. So they live in the lymphatic system. The lymph is just fluid that's filled with immune cells, T cells, B cells, innate immune cells that we haven't covered yet. The fluid that runs through the lymph is actually the exact same fluid that runs through your bloodstream. So that fluid, the fluid in your bloodstream leaks out when it gets to your capillaries. Capillaries are really porous, so you lose a lot of that liquid from your bloodstream in your capillaries. So it leaks out into your tissues, and then it's picked up by the lymphatic system. So it's the same fluid that travels alternating back and forth between the bloodstream and the lymph. So when that fluid is in the bloodstream, we call it plasma. When that fluid is in the lymphatic system, we call it lymph. There's different structures in the lymphatic system that carries lymph. We have lymph vessels, lymph nodes, and lymph tissue. So here's just a diagram of the lymphatic system. So lymph vessels are these green lines. They're vessels kind of like blood vessels, except they're open-ended. So your blood vessels are circular. It's a closed loop. So your blood goes out from your heart, loops back to your heart, goes up to your lungs, loops back from your lungs, goes out to your extremities, loops back. So it's a closed system. The lymphatic system is not closed. It just, all the fluid that leaked out in the capillaries from the blood circuit gets picked up from the capillaries and returned to center. And that's it. It's a one-way system. So the fluid is picked up into the lymphatic system where all the white blood cells, it's washed of pathogens before it's returned back to the heart. So here it's returned back to the heart. So lymph nodes are these little bean things. Lymph nodes are just, they've got a lot of, so they're openings. So they're where the lymphatic vessels widen. And they're filled with fibers that act sort of like filters, and they catch debris like pathogens that are floating through. So it's got these fibrous filtery things, and also a lot of white blood cells hang out there on those fibers. So debris gets caught, and phagocytize that debris and activate an immune response if you need it. And then what do we have? Lymph tissue. So we have lymphatic organs, which we sometimes call lymphatic tissue, and there's several different kinds. So bone marrow is considered lymphatic tissue or lymphoid organ, because in the bone marrow, that's where all your blood cells, including your white blood cells, begin their lives. The stem cells for every kind of blood cell, red blood cells, platelets, white blood cells, all those stem cells are in the bone marrow. So white blood cells all start their life in the bone marrow. So that's a lymphoid organ. The thymus, we talked about how T cells finish maturing. So they start in the bone marrow, and then they go to the thymus to complete their maturation and for thymic deletion, to get rid of all the T cells that could attack your own body. But then you have these other lymphoid organs like your tonsils. Your tonsils are larger than lymph nodes. There are places where they surround your mucus linings. So you have a lot of lymphoid organs around your mucus linings because mucous membranes are really vulnerable to infection. So you want these larger organs filled with lymph, filled with white blood cells to help fight infection for your most vulnerable tissues. Your spleen is part of filtering your blood, also is considered a lymph organ because a lot of your white blood cells hang out there and destroy old damaged red blood cells as well as pathogens. And I want to focus on Peyer's patches. Peyer's patches, there's only one shown here, but they actually surround your intestines. Your intestines are also very vulnerable to invasion because you have massive amounts of microbes in there. Everything you eat that gets digested, it can be filled with microbes that could cause various styles of disease. In addition to that, you have this huge microbiome that you need to create boundaries with, because any one of those, so when you die and your immune system stops working, the reason you decompose is because you are full of microbes and most of them are living in your gut. As soon as your immune system isn't holding them back anymore, they start digesting you. So yes, they do a lot of good for you. Some would even say that they're essential for proper health. But your immune system is actively creating proper boundaries and managing that microbiome appropriately. And so those white blood cells have an important job around your intestinal lining. And so these Peyer's patches, I want to emphasize them because I'm going to talk about them a lot when we get to the microbiome chapter. So here's the Peyer's patches in more detail. So Peyer's patches are lymphoid organs that surround the intestines. And the reason they're important is because each Peyer's patch is small, but collectively they make the largest lymph organ of the body. 80% of your immune cells are residing in Peyer's patches at any one time. 80%. So the majority of your white blood cells at any moment are in your Peyer's patches surrounding your intestine. Okay. So these are very important to your body. So you'll notice that this is your intestinal lining. These are your intestinal epithelial cells. They're wavy on this side because that increases surface area for absorption of nutrients. The food stuff is going to be up on top of them here. And so the nutrients are being absorbed through these cells and released on the other side of your body. So you're absorbing nutrients. But you'll see that this cell here, and then down here is the Peyer's patch, right here. You'll see, and it's filled with these white blood cells that are preemptively generating immunity to all of the microbes in your gut. Here is a cell that looks different. This pink cell is not wavy at the top. Its job is not to absorb nutrients from your food. It is called an M cell. M cell. Now, every year I get students confusing M cells with M proteins. We talked about M proteins in microbial mechanisms of pathogenicity. M proteins are proteins on the surface of Staphylococcus species, and they are virulence factors. M cells are part of your body, and this is what they are. So the job of the M cell, they always sit on top of Peyer's patches, and their job is to endocytose antigens from microbes and also full microbes and release them in a controlled way to the Peyer's patches with all the white blood cells beneath. So what that does is it takes, this gut is just filled with trillions, literally trillions of microbes. Your Peyer's patches are giving a slow release of those microbes into the Peyer's patches so that the white blood cells can generate immunity to them preemptively. Because if at any moment any one of these microbes gets out of the gut and into your bloodstream, that would be bad. Even if they're friendly microbes, even if they're microbes you want to have, even if it's E. coli producing vitamin K for you, you don't want it circulating in your bloodstream. You want it in your intestine. You don't want it inside your body. So your body needs to generate immunity to every single one of those trillions of microbes in order to create a healthy boundary. They belong here, they don't belong here. So your white blood cells have immunity to every microbe that lives in your intestinal lining. The job of these M cells is to help with that. Bring in the antigens and the microbes slowly, expose it to the B and T cells beneath, let them generate those memory cells and those antibodies, and that helps maintain a healthy boundary between you and your microbiome. And here's one last picture of the M cells. So here's your intestinal lining. It's very wavy. Here's all these intestinal epithelial cells that have those wavy surfaces for nutrient absorption. And then here's a Peyer's patch filled with white blood cells. And here's the lymph node that feeds it, part of the lymphatic system. And then here's the M cell on top. Microbes and digesting food is up here. White blood cells are here. And then your bloodstream is all mixed up in this as well. So this M cell is slowly bringing in antigens and microbes from your gut, exposing your white blood cells to them so that you can generate those memory cells to help protect you from your own microbiome. Usually they don't get through. This is a really good barrier. We'll talk about it when we get to the microbiome chapter, but these are really tightly packed and almost never does something get through. But if there is a wound or inflammation, it can increase vulnerability to invasion, and you just want to be ready just in case. So that's what those M cells at those Peyer's patches are for.